My name's Ken Sprankle, and I'm a fishery biologist with the U.S. Fish and Wildlife Service. And what we've seen with our data for the Connecticut River is basically since the early 1990s, around 1992, marine survival rates based upon how we examined our data and tracked fish numbers and did analyses and assessment work. Since around 1990, marine survival rates have been anywhere from 1/7 to 1/10 what they were prior to 1990. And that's been consistent. It's not changing. So it's been since 1990, so you're talking over 2 decades now. And that's why the federal government said, there's no information, no data to show that anything's going to change on this, and so we're no longer going to work on restoration. And the states kind of reluctantly agreed with us. They couldn't do it without us. So marine survival rates, even in the face of reduced commercial fisheries, there are other just very large-scale unknowns that are occurring that have seriously impacted survival of fish in the marine environment. There are things that you can control, and there are things that you can't control. So the things under management control, we do the best we can on them. And in some areas, I think we can do a very good job. So the genetics, right now in Maine, and we did it in Connecticut, we genotyped every fish that came back. And so we were selecting which fish to mate with which fish to ensure no inbreeding or outbreeding occurred, to maximize the genetic diversity, to maximize the opportunity to not lose unique alleles, that at some point, if one fish spawned with another or some fish out in that environment survived, that could have been very important. But that takes time, and there's no book on it. There's no way of knowing how, when those things operate. All you can do is, use the best science, the best information, the best approaches you can, and hope that you're minimizing potential negative effects from your hatchery practices. Because the hatchery practices were the only reason there are any salmon left in the United States of America. I mean, without hatcheries up in Maine, and certainly, where the restoration where there were none. So when you're talking about dams, I think for the Connecticut River program, one of our most important hurdles relative to dams were to ensure that the fry that we were stocking, as a strategy that evolved over time through evaluations and research, that really came into primary play, I would say, in the early 1990s. That's when we were really kind of hitting our stride and saying, okay, this is the approach, and we're going to need to stock out 10 million fry throughout the Connecticut River basin. So this huge basin, we're talking the upper reaches of Vermont and New Hampshire, way up there above lots of dams. We need to ensure that those smolts that are produced from that can survive that out-migration. Well, even with the best technology and best practices and evaluations we've conducted, and they encounter a dam, we'd like those dams to be invisible to them. And of course they're not, but you'd like to have those fish not be delayed. And by delay, through research that we've been involved with and have conducted, the smolts have a very limited time window where they're undergoing these physiological and behavioral changes. It's triggered primarily by photo periods. So if you have, again, a fish that's stocked well upstream in a basin, and not only are there risks associated with the turbines, but there are potential issues associated with delays. So with that dam there, they encounter the dam, they're trying to figure out, well, which way do I go? I don't like it over here, I don't like it over there. If they spend a couple extra days at each dam, if they spend 2 days, maybe they spend 3 days. Well, if they have 30 days to get out to the ocean, and they have to encounter 5 different dams, if they're late in their run for any number of reasons, maybe they just started later, that window can be really tightened, and they may find themselves entering the saltwater environment with their body, with their physiology no longer prepared to make that transition to saltwater. So there are special cells located on the gill lamellae. They're salt pumps, are what they are in the most basic sense. The salt pumps become more developed and activated, and so as they're migrating downstream, it's actually called sodium gill ATPase is the actual enzyme that's occurring there. But that's going to allow them to transition from that fresh water to salt water. If they're not ready for that, they'll die. And so what happens is, if it's too soon, they die. Of course, if it's too soon as they're going downriver, they can choose to kind of go back and forth and take their time. If they find themselves well downriver, ready to enter the river, and that whole time window has passed and everything's gone, they're out of time. And they're in habitat that is not suitable for juvenile salmon. So not only are there the immediate potential effects from the turbines and shear stress and physical impacts, but the delays were always also another very important concern.
Baby, bring me home to bed.